Hi, I'm Mr. Baker, and this is Unit 9 Protein Synthesis. We're going to cover DNA review, the intro to RNAs. We'll talk about transcription, translation, the genetic code, and finally, mutations. A refresher on DNA structure that are built out of nucleotides. Those are the building blocks of DNA RNA, and it consists of a 5-carbon sugar. In the case of DNA, it's deoxyribose, a phosphate group, and one of the 4 nitrogen bases. They have a sugar phosphate backbone that you can see here with a sticky sugar, a phosphate, and a base. The bases come in 2 main flavors, the purines and the pyrimidines. Purines have a double ring. Those are adenine and guanine, or A and G, and the pyrimidines have a single ring, cytosine and thymine. I keep them in my head together with the pyrimidines with a Y, and cytosine has a Y, and thymine has a Y. They are base paired together, connected by hydrogen bonds, and you need to memorize that A always binds with T and C always binds with G when we're talking about DNA. A set of 3 bases that codes for a specific amino acid is something called a codon. And you will do something for homework, just like this. We'll give you a piece of DNA, give you the RNA, they give me the codon, and what it means for the amino acid. You need to memorize that it's an anti-parallel orientation, where one strand runs from 5' to 3'. The other one is upside down, or 3' to 5'. Those 3' and 5' are just ways that biochemists number the particular carbons in the atom. You just memorize it. One's 5 to 3, and the other one's 3 to 5 as you look at them. This is called anti-parallel. They are also complementary, which means that if you have one, you can determine the other, because A will bind to T, T to A to T, and there it is filling in for you the complementary matching DNA strand underneath the one that I have given you in the notes. DNA provides all the instructions, and these instructions are into segments that we call genes. Each gene provides information for making a protein, which carries out some type of thing that the cell needs it to do. RNA is different than DNA, and first, the sugar is ribose, not deoxyribose. Second, it has uracil instead of thymine, so we're switching out one of the bases. It's a single strand, not a double strand, but it has structures called hairpin loops where it can fold back upon itself. There was a video for you to watch. There is a diagram showing you the difference of the single strand versus the double strand. There are 5 types of RNA, including pre-RNA, which is newly transcribed, snRNA, which are small nuclear RNA, SNRPs, and those are the RNA scissors. mRNA, M for messenger, rRNA, ribosomal RNA, you find that in the ribosomes. tRNA is a transfer RNA, and that's what's going to carry the amino acid to the ribosome to be added to the growing protein chain. They have a clover structure, and they don't have codons. They have the match to them, the complementary match. That's called an anticodon. Here are your 3 types of RNA. Top one has the ribosome. It's the large orange blob. The middle one has the messenger RNA, and that's the strand near the bottom. And the tRNAs are those goofy-looking green things that keep popping in. Shout out to the Amoeba Sisters for putting out such a good GIF. tRNA has the anticodon. It's going to bind to the codon. You have a different tRNA for every amino acid, and it gets its name tRNA because it transfers amino acids to the ribosome during a process called translation. More about that coming up. Here is it in its cloverleaf structure, and in a 3-dimensional model, it looks like a bent firecracker. Another picture of the bent firecracker that's more technically correct. Ribosomes, don't forget, those are the machines on the assembly line. They're made of RNA and proteins, and there are 2 pieces to them. There's a picture. The larger piece is in light blue, and the smaller piece is in, what is that, kind of yellowish. That's your quiz for 9-1. Let's move on to transcription and translation. Protein synthesis, and we're going to talk about transcription, and that's making a copy of the DNA into mRNA. And then translation is going to be protein synthesis, where the RNA, the mRNA, and the tRNAs all come together to build a protein, which is an amino acid chain. First, let's talk about transcription. There are 3 steps: initiation, elongation, and termination. You need to know the players. The big player here is RNA polymerase, or you can call it RNA pol. It makes the RNA copy of DNA. You have transcription factors, which help the RNA pol find a promoter site. A promoter site is where the RNA polymerase binds, and it's always at the beginning of the gene. And you're going to have something called a termination site, which is always AAUAA. This is where RNA polymerase will fall off the gene being copied. It's always at the end of the said gene, hence termination. The first step is initiation, and you got to have a promoter site. And RNA polymerase is going to bind to the promoter site on the gene. There's a picture that gives you some idea of what's going on. It's more complex than we present. You just need to know that promoter site this is the area where RNA polymerase is going to bind. Elongation, now you're going to build the RNA, and you build it in the 5' to 3' direction. And you're going to add nucleotides complementary to the DNA template. Here's a video that I've shown you in class. We're not going to take the time to watch it here. Termination is when RNA synthesis ends, and it's going to reach that termination signal, which is AAUAA. And then you had a video to watch. And then we're not quite done with the RNA yet because it needs to be processed. And this is called splicing. RNA processing, the just transcribed RNA is called pre-RNA. And we're going to put a cap on the front and a tail on the end. It's a guanine cap. It goes on the 5' of the front end, and a poly-A tail, meaning a whole bunch of adenines. Poly, many, A, adenines. It's added to the end. And it can be from anywhere from 20 to 200 adenines. You need to know cap and tail go on. And these function in keeping the RNA from being degraded or falling apart, meaning that they protect the RNA. You just invested all this time and effort in building an RNA. You want to keep it around. Here's a video that you should have watched at home. Now we need a splicing because we have pieces of RNA that are in there that we don't need. Those are intruder pieces, and they're called introns. We got to cut those out. What we don't cut out and what's left behind are the exons, and those are the coding regions. And doing all this is called splicing. And what does it? The RNA scissors, or SNRPs, which is probably the coolest biological term. So here's a diagram where we've got the two green dudes are the introns. We need to cut those out and put a cap on, put a tail on, and the introns are gone, and we left with 3 exons. Another video showing you this in great detail, and a couple of pictures showing it in great detail. So for the pre-RNA, we're going to put a cap, a tail, we're going to cut out the introns using the SNRPs, we're going to leave the exons, and now you have a functional mRNA, and it's protected from falling apart too early. RNA seems to have evolved first because they can perform other functions, and some of those are called ribozymes. It's a mashup of ribosome and enzyme. So let's review. The first step of gene expression is transcription. RNA polymerase is going to be a player. It's going to produce a complementary RNA strand. You have A to T in DNA, but you have A to U in RNA. Transcription has 3 steps: initiation, elongation, and termination. And here's a nice little table we put together. Transcription, where nucleus. Is DNA involved? Yes. RNA, which ones? Pre-RNA and mRNA. What's our end result of transcription? We're going to build a molecule of mRNA. Translation, where? In the ribosomes. More importantly, is DNA involved? Nope. Which RNAs are involved? mRNAs, tRNAs, and rRNA. And the end result, we're going to build a protein or an amino acid chain. That's your quiz for 9.2, including transcription, the initiation, elongation, and termination, the processing and splicing, and don't forget the terms introns, extrons, SNRPs, and what cap and tail we put on. Study hard, and we'll see you next time.